So we do start off with a few questions today to it was kind of like maybe a 3-day weekend. Let's warm up our brain cells again. Alright, so let's start off with the first question of today. Okay, so which of these yields the most net calories in non-starvation conditions? So this means to your day-to-day. So you're not starving, you're eating your typical caloric intake. So say you eat some of this, which is going to give you the most calories? And notice I put 1 gram. So again, it's all the same mass, but which gives the most calories per mass? Okay, let's see the responses. Most of you said fatty acids, and those of you who said fatty acids are correct. And why is that? Again, if you look at the back of your nutrition label, you see grams and calories as well. So you're able to convert this, but how are you able to convert from grams of these macronutrients to calories? Well, this is our next question. So match each type of molecule with your energy yield per gram. So this is a sorting question. Yeah, so ethyl alcohol or ethanol or drinking alcohol, that's just more FYI, but these are the correct responses. So the ones I definitely want you to focus on are amino acids. So again, these are like from our proteins. This is why we get 4 kilocalories per gram. And fatty acids and lipids, we get 9. So this is again why fats have more calories per gram. And glucose, which is an example of carbohydrates, that's why there's 4 calories per gram. Nucleic acids, again, this is why they would have the least, because they're not on the nutrition label, because unless you're like really starved and your body is just trying to get carbon from somewhere, you're not going to metabolize nucleic acids. So this is again, this is how you're able to get that calories per serving and total calories from a nutrition label. And yes, I know sometimes the math doesn't work out, and that's kind of like due to rounding errors and other parts that are kind of problematic with nutrition labels, but this is how you're able to get the total calories from some sort of meal or some sort of food that you're intaking. And before we get on to that, now one final question is for today. So nutrients cannot be made by your body. They can only be catabolized and turned into energy. Is this statement true or false? So the answer is, and most of you, well, what are the responses? So most of you said true, most of you, but some of you said false. Now, what I'm getting at here is anabolism versus catabolism. Can you build up these macronutrients again? Well, the answer, believe it or not, is that you can make certain nutrients. And we did cover an example of this earlier in the semester, but hopefully we have enough time to talk about this. So yeah, the answer is false. You can create nutrients in your bodies, but not all the nutrients you need, but you can create some of the nutrients you need to maintain your cells, and some of these macromolecules, you can make some. Back to business. So let's talk about metabolic pathways. And each node is some sort of chemical. Each line is something describing a chemical reaction. So this is Martine's version of kind of representing metabolism, but in a less complex fashion. But what we have here are the 3 major types of macronutrients and how they're broken down and converted into usable sources for metabolism. So today we're going to talk about carbohydrates and glucose. So the thing about this is, like, can you create new glucose? Can you create new, how do you break glucose down, and how do you make new glucose molecules? Alright, so then glucose. Do I expect you to memorize the exact chemical structure of glucose? No, not for this class. That's something you'll probably have to, you'll definitely have to memorize for organic chemistry and biochemistry. But glucose, let's talk about its energy content. So glucose has less energy content than fats, but it's easily converted into ATP. That's why, even though it's only got 4 kilocalories per gram compared to 9 kilocalories per gram for fats, it's easier to use and burn and convert into ATP. So we have to talk about how we convert glucose into energy. This might be reviewed to you if you've already taken AP biology or basic biology, but the key to this is just metabolism and converting these macronutrients to ATP are carbon atoms. So again, I don't expect you to know the structure of glucose, but there are 6 carbon atoms. So whenever you see this kind of like skeletal structure of a molecule, if it doesn't tell you what atom it is, it's typically carbon. So carbon is shown here with all these angles right here where there's no specific atom. The reds are oxygen, the grays are hydrogen in this example. But this relates to this equation that might be very familiar to you. So what we have is glucose being combined with oxygen and then metabolized into carbon dioxide, water, and energy in the form of ATP and heat. So this is the classical cellular respiration or aerobic respiration um, equation. But how do you convert glucose to ATP? Well, there are several steps. So glycolysis is the first step, and this is why carbon is a big deal, because it deals with like how you metabolize. Now, do I expect you to memorize all these compounds right here? No, that's more for your upper level classes, and if you go into med school, you gotta memorize this anyway. But for this class, I want you to appreciate what the starting product is and what happens to it along the way, and the ending product, and especially as it relates to ATP, because that's one of the forms of energy that we get from metabolizing glucose via cellular respiration. So glucose again has 6 carbon atoms, and this molecule is also glucose. And if you go to organic chemistry, they'll talk about the ring versus the linear structure. But whether it's a ring or a linear structure, it has the same molecular formula, and you have 6 carbon atoms. Now this is the linear this form right here, the ring structure, is just showing that there's an extra bond between this oxygen atom and this carbon right here. But the thing is that this glucose can fluctuate between these two structures all the time. But the linear structure is useful in terms of how you metabolize glucose into energy. So glucose has 6 carbons, and again, I don't expect to memorize these steps I'm going to say right here. So 6 carbons, and what happens is that ATP is used to add phosphate groups to the structure of glucose, and then now it becomes a different compound. Again, I don't expect you to know what compound it is for this class, but the thing about adding this phosphate makes this overall chemical structure unstable. And unstable meaning that its bonds are weaker and it's more prone to being broken. So these bonds are more easily broken, and it breaks like this, but now we get 3 carbon, so we get 2 molecules, but they have 3 carbon each. So these 3 carbon fragments undergo changes, and what changes do they undergo? Well, they undergo changes like this. So they undergo metabolic changes, and they start shuffling their atoms around. But even though their atoms are shuffling around, they still have 3 carbons. Eventually what happens is that these 2 molecules with 3 carbons, they end up being identical. So they're not identical at first, but they eventually become identical, and these molecules are called G3P. And this is more like if you're taking like the MCAT or going to med school, this is like common question. But what I'm getting at here, they start off, you take 6 carbons, you break them apart, you shuffle them around, and now you have 2 molecules that have the same structure. Now what happens to these 2 molecules? More chemical reactions. And these chemical reactions, you start losing atoms, you start shuffling these atoms around. You still have 3 carbons, and eventually both of those molecules become something called pyruvate. So now from one glucose molecule, you have 2 pyruvate molecules that have 3 carbons each. So that's what I want you to know. 6 carbons in glucose, break it apart, now you have 2 identical pyruvate molecules. Okay, so what we have here, that's the overall process of glycolysis, but notice that in the first step we need to use up some ATP, and this is an important part because why? It tells you in this overall process you actually gain 2 ATP molecules, but you actually made 4 during the process, but you also used up 2, so the net gain is 2. So glycolysis occurs in the cytosol, so that's the liquid inside your cells, not in the mitochondria. So that's one important part to get from this. So what is the initial reactant that goes into this process? You take glucose, and then you break in 3 carbon molecules. You shuffle it around, and again, these are the steps. I don't expect you to know the shuffling part. But what comes out of it is 2 pyruvate molecules, and these pyruvate then end up in the mitochondria. So again, glycolysis does not occur in the mitochondria, it occurs in the liquid inside your cell. Now the ATP yield, so some ATP is generated, but in the beginning we used up 2 ATP molecules, but you generate 4 ATP during that process. Where it occurs, that's I think a more med school or upper level biochemistry class question, but the thing is that you're generating ATP overall. So you have 2 net ATP being generated from glycolysis. So again, the glycolysis also produce something called NADH via this process called reduction, which we'll hopefully get to by the end of this class. So again, here are our learning goals and key points. And so where does this pyruvate end up? So we took glucose, now we have 2-pyruvate. What you're going to do is take this glucose that you made into pyruvate, which are these 3 carbon chains. So pyruvate has 3 carbons, and then you're going to convert it into something called acetyl-CoA that has 2 carbons. Now this actually occurs like before you actually enter the citric acid cycle, which we're going to talk about. But this is a step that's important to lead up into the citric acid cycle. So acetyl-CoA is when you take that 3 carbon pyruvate, but what you do is you lop off one of the carbons. In fact, you're going to lop off this carbon right here, which is CO2. That's carbon dioxide. So this is where one of the sites that we produce carbon dioxide in that cellular respiration equation. So what you do is like the carbon dioxide, it's excreted via your respiratory system, but now you're going to add something called CoA or something called CoAsh because there's a sulfur hydryl group. But what this acetyl-CoA is going to anchor these 2 carbons, so this acetaldehyde group right here, to coenzyme A. Now, this acetyl-CoA, what it's going to do is carry these 2 carbons into the citric acid cycle, or commonly known as the Krebs cycle. You probably heard that sometime during your education. Now, with this acetyl-CoA, so what is this? Is it really that small? It's showing this CoA because the actual molecular size is really big. So CoA is coenzyme A, and if you drew out all the atoms and bonds, it would look like this. So yeah, this is the acetyl part, but this part is the acetyl, and this coenzyme A right here is all of this over here. So that's why they're representing it as CoA versus just like showing the entire structure. So what's going to happen is that this pyruvate, when you convert that, you lop off that carbon dioxide, but now you're just swapping in this coenzyme A for that carbon dioxide over there. Now, what you have over here is now you end up with a 2-carbon compound, or 2 carbons from the acetyl group. And now this CoA allows your cells and enzymes to manipulate these 2 carbons and use them in chemical reactions. So I like to draw an analogy between this, like this acetyl group is very, very small. So it's actually really, if you had it by itself without anything attached to it, your cells would diffuse throughout your cells, and your cells and enzymes would have a hard time holding on to it. So what your cells are going to do to make it easier is attach it to a bigger molecule. So that's the purpose of coenzyme A. So it's kind of like my friend does, he makes a lot of jewelry, and these jewels are very small, but is it easy to manipulate a very small jewel with your fingers like this? Not really, right? So what they use is like a jeweler stick, or I think they call it a dab or something like that. But what they do is take a jewel and they attach it to using wax and a larger stick to help it make it easier to set these jewels, or use these small stones that could easily fall away and roll off somewhere. So by attaching it to a bigger molecule, acetyl-CoA, or attaching an acetyl group to a larger molecule, makes it easier for an enzyme to kind of grab onto this overall structure and manipulate this tiny, tiny, tiny acetyl group. So that's why we have this acetyl-CoA. Now, where does acetyl-CoA end up? So now it ends up in that infamous citric acid cycle. So from acetyl-CoA, you add acetyl-CoA to oxaloacetate and you make citrate. And then it undergoes these multiple chemical reactions where citrate becomes isocitrate, becomes alpha-ketoglutarate, becomes succinyl-CoA, becomes succinate, becomes fumarate, becomes malate, becomes oxaloacetate. Yeah, so this is like a classic thing if you're taking the MCAT, and I'm pretty sure the NCLEX has it as well. This is like a classic test question. Now, you might be wondering, okay, is it useful to do this? Like, this is a wise cycle. You just go back to step 1 as when you add on another acetyl-CoA. Now, will you ever need this? So I don't want you to tell your other professors, like, especially if you're taking biochemistry or any advanced classes, that yeah, you won't need this in clinical practice. But why is it like constantly tested all the time? Yeah, so I mean, even the only time I've used it in my career, the only time I've used the Krebs cycle is to take tests about the Krebs cycle. So this, and every time it's like, okay, I have to kind of refresh it because I don't really use it. But what's the whole purpose? And the thing is that This is the Krebs citric acid cycle is pretty much scientific canon. So this is like nothing's really going to change with this over here, and the enzymes have all been discovered. The parts of the citric acid cycle have been discovered. But this is what I want you to get from it. So this is also known as the TCA cycle, the tricarboxylic acid cycle, so many names for the same cycle. But it occurs in the mitochondria, and this is why the mitochondria are the powerhouse of the cell, because this is an essential part of converting many of the macromolecules to energy. So it's fueled by acetyl-CoA, so not pyruvate directly. Because again, pyruvate has 3 carbons, and this oxaloacetate actually has 4 carbons. Notice the citrate has 6 carbons, so you need to add, what's 4 plus 2? That would be 6, right? So you can't add pyruvate, you actually have to lop off the first carbon first. But the acetyl-CoA is going, every molecule of acetyl-CoA can do 1 full cycle of the citric acid cycle. Now, along the citric acid cycle, different things are produced at different parts of the citric acid cycle. So you produce this mysterious chemical we haven't talked about called NADH, and also something familiar called carbon dioxide. You also produce something called GCP, and it's actually converted to something else called ATP. So you produce carbon dioxide, you produce ATP, but you're also producing these other molecules called NADH over here, NADH and FADH2. They are different, but they serve a common purpose. So this is the part I want you to know for the citric acid cycle. Do I think it's worth knowing all the enzymes and intermediates practically? That is more, I think, just something, the standard that people test just to see like how much knowledge you can get in your brain. But what I think the practical part is this acetyl-CoA is very important for driving the citric acid cycle. But before you even get to acetyl-CoA, you lop off that carbon dioxide and you produce NADH. Now, okay, so what we have here is like a 4-carbon molecule here. That's oxaloacetate. So it combines with the 2-carbon acetyl-CoA, and that makes that first part of the citric acid cycle citrate. So that's where the TCA cycle comes from. Now, citrate, what happens is that this 6-carbon molecule loses a carbon. So it becomes a 5-carbon compound, and this 5-carbon, so how do you lose a carbon? You lose it in the form of carbon dioxide. And the interesting thing is that this, as you lose this carbon dioxide, this also produces yet another molecule of NADH. Now this 5-carbon molecule is going to become a 4-carbon molecule, and how does it do that? It has to lose carbon. So how do you lose the carbon? You lose the carbon in the form of carbon dioxide. So notice that what's also being produced, what also comes out of this reaction is yet another NADH molecule. Are you seeing a pattern here? You see that every time you lop off a carbon dioxide in this overall process, you're generating NADH, and NADH is important. I'll get to that. Now the 4-carbon, along the way, going back and looping back here, you do generate ATP. You actually generate GTP directly first from this citric acid cycle, but your cells are easily, they can exchange GTP for ATP 1-to-1. So it doesn't generate ATP directly, it's due to GTP. That's why I say ATP by GTP. Now, 4-carbon, as a different steps along this 4-carbon, it's going to shuffle, it's going to maintain its 4 carbons as its central structure. But as it's shifting around the different atoms around this 4-carbon basic structure, other things happen. You have all these protons and hydrogen atoms being released from the structures, and it's being absorbed by and converted into NADH and FADH2. So these are these molecules. So the major products is not ATP yet. You do have some ATP, but the major products of the citric acid cycle are these NADH and this FADH molecules. So this is what I want you to get from the citric acid cycle. You involve carbon and the release of carbon by, again, popping off carbon dioxide from pyruvate before you get to the citric acid cycle. And once you add acetyl-CoA to the 4-carbon and make citrate, you pop off 2 more carbons, and this is where you generate NADH and also carbon dioxide. So per pyruvate, you have 3 carbon dioxide, you have 1, 2, 3, 1 ATP per cycle, and then 4 NADH and 1 FADH2. So that's what I want you to get. So what goes into the citric acid cycle? And this is an intermediate step, but let's include it here. So per pyruvate, you get all of this. Now, how many pyruvate do we get per glucose molecule? Again, we have like 2 pyruvate per glucose. So if we said this was per glucose molecule, all you do is multiply this by 2. Alright, so NADH and FADH2. What are these mysterious molecules? Well, these are, the whole purpose of these are to store electrons released by the citric acid cycle. And these stored electrons, and actually protons as well, can be used in making ATP. So you don't have ATP yet, but this is the way you're storing energy from the citric acid cycle. It's through this NADH and FADH2. Do I expect you to know these structures? No, again, some of you are still taking chemistry and finishing it up, so this is more for biochemistry. But this is what I want you to get from that. So we have to talk about oxidation and reduction, and again, your chemistry classes will talk about this more in detail. But what's oxidation is that we have a chemical compound and then you lose electrons from the compound. That's called oxidation. Reduction, now this is where it's a little tricky. Even though it sounds like reduction sounds like you're reducing something or you're lessening something or getting rid of something, you're actually gaining electrons. So reduction refers to the charge of the overall molecule. Because again, electrons have a negative charge, so it's not talking about losing electrons, it's talking about reducing the overall charge of the molecule. So this is reducing the charge, so like with NAD, when we reduce NAD to NADH, you're reducing the charge, you're going from positive NAD structure right here, that's a charge of +1, and you're making a neutral NADH. So you're going from +1, +1 to 0. So that's a reduction in that number, right? So this is what's happening with this here. And yeah, so in both of these, when you reduce these, what's happening is that you're adding electrons to the overall structure. Now, with NAD, it's pretty intuitive. So what you have with this is NAD by NAD plus. It has a +1 charge. So this is the oxidized form, and it has a positive charge because, again, +1. But NAD, what happens when you reduce it, you add a proton, but you also add 2 electrons. So what's 1 + 1 minus 1 minus 1 again? That would be 0, right? So again, or what's 1 + 1 plus -2? That would be 0, so that's why NADH has a neutral overall charge. So there's no overall charge with NADH, but even though you have no charge right here, what did you pick up along the way? You stored a proton and you stored 2 electrons. That's the main thing I want you to get from this. So you stored protons and electrons. Now FADH and FADH2, is there an atom called AD? This is like one of those where, it's like acetyl-CoA. The structure is actually way bigger. I don't expect you to know the structure, but what I want you to appreciate from this is that FAD, what you do is you add protons and you add electrons, but you're adding equal amounts of both. So again, protons have a positive charge, electrons have a negative charge. So what you're doing is adding 2, but you're also subtracting 2. So what happens is, to overall charge, you have FADH2, but it has a neutral 0 overall charge. So, but does that mean nothing happened? No, you absorbed you absorbed 2 protons, and you absorb 2 electrons while going from FADH to FAD to FADH2. So this is why it's a FADH2, because of these 2 hydrogen atoms. But again, this is why there's no net overall change in the charge, because you also added 2 electrons in the process. But either way, what happened is that in going from NAD plus to NADH, you gained electrons. Going from FAD regular to FADH2, you gained electrons. That's the main message I want you to get from this. So oxidation and reduction, like actually I learned this from a previous student. I like this one. Oil rig is a great mnemonic. So oxidation is losing, reduction is gaining. And what are you losing and gaining? You're losing electrons, you're gaining electrons respectively. Now NADH and FADH2, this is not ATP yet. So, but what we did is, through the citric acid cycle, we generate a bunch of this NADH and FADH2. So this has energy, but not yet in ATP form. So I like to think of it this way. It's like glucose, and not encouraging money laundering, but remember how I said like energy is kind of like currency and cash and money? So glucose is like, it has a lot of value, it has a lot of energy stored up, but you have to convert it into something that's useful. Again, you can't just like take your gold chains and buy something from the convenience store, right? So how do you go from here to here? So what you're going to do, one way you can do that is convert things into different forms of currency. So maybe for NADH and FADH2, you're converting your glucose, and maybe you have like one of those gold buyers. They say, well, we can't give you cash up front, but we can give you to you in the form of a charge card. So charge cards, they still have some sort of value, right? They have some monetary value, and you can convert them or use them into ATP or energy eventually, or money eventually, right? And cash. So glucose is just different forms of energy. That's what we're getting at here. So this NADH and FADH2 is an intermediate step in converting the energy from glucose into ATP. But what is it going to do is store electrons and this protons and energy for later. Now, where does this NADH and FADH2 go? Well, it's going to end up in the electron transport system. And this is where so what you're doing is like building up all these gift cards, and now you're finally going to cash them out and finally get what your final product is. So now you're going to take all this NADH and FADH2 and finally convert that into ATP. So that was the citric acid cycle, and these are the key points and learning goals from that. Now let's talk about the electron transport system, and what we have here is like, okay, all the NADH and FADH2 go into this electron transport system. Now, what are they doing there? Now, do I want you to memorize this, all the complexes and this overall anatomy and histology of the mitochondria? No, I don't expect you to know these complexes. That's more for biology and biochemistry. But what I want you to know is where NADH and FADH2 plug into this. So here we have NADH and FADH2. And they actually start off in this, what we call the electron transport chain, or sometimes called electron transport system. And what's going to happen is that these are going to now take their electrons, and they're going to put it into this system. So you're going to lose the electrons here and go into these little proteins that are found in the membranes of the mitochondria. And actually, let's take a step back. Look at the mitochondria. Compared to the other organelles in your cells, they actually have a double membrane. And that's what you can see over here as well. So there's an outer membrane and an inner membrane. But what happens is that you have all these proteins on the inner membrane, and NADH and FADH2, they donate their electrons to these proteins. So these electrons, these are like the power that drives this electron transport chain. So what is it powering? It's powering the transport of protons into the space between the two membranes of the mitochondria. So this space is called the intermembrane space. So this is the whole purpose of those electrons. They are moving these protons into this area over here. So this area is going to be actually have a very high proton concentration. And remember that protons are acids, right? So with a high proton concentration, you have a very acidic intermembrane space. But what happens is if you build up all these protons, is that what you have, so yeah, here we have FADH2 electrons adding even more protons to this system, is that these protons, if you open up a channel that allows these protons through, they're going to flow from a high concentration of protons to a lower concentration of protons. So the interesting thing is that this built up concentration of protons is actually an energy source. And there's a special enzyme called ATP synthase. This is going to take the energy from protons moving through it, and every time, or actually I should say, like during this process, it generates ATP. And there's a little debate about, okay, what the exact ratio of protons to ATP is. Don't worry about for this class. That's more of an advanced biochemistry topic. What I'm getting at here is that what is the purpose of NADH and FADH2? They are going to use electrons to move protons into the space between the two membranes of mitochondria. And this, by doing this, builds up all these protons, and these protons, through this ATP synthase, which allows these protons to leak through and move from the space back into the innermost part, the mitochondrial matrix. As these protons move back here, it's going to generate ATP. Now, if we look at this closer, this part right here, notice that every time these protons come back, you're generating ATP. And again, don't worry about the exact ratio. But what happens is that these protons, they don't just flow around freely. They actually combine with oxygen. So this interesting thing is that this oxygen combines with these protons, and what do you end up eventually with is H2O, or water. So remember that cellular respiration equation, how it takes oxygen and combines it with glucose and then makes carbon dioxide and water? This is where the part that water goes. So you're consuming that oxygen at this final step and you're generating water. So that's why oxygen goes into that equation and we get water coming out. This water is specifically called metabolic water, but that's like something I don't really focus on in this class. The whole purpose here is like, okay, you use those electrons to move this whole system. This overall process is actually called oxidative phosphorylation, because why? You're actually adding phosphate to ADP, and then you're finally generating ATP. So the thing about this is that, or here's an analogy I like to use, it's kind of like these little hydrogen atoms, these protons are kind of like workers right here. So what they're doing is they go to work, so here's what and when they go through work, they have to go through an electronic gate that requires some sort of electronic swipe and activity. So NADH is kind of like this. It's kind of like that electronic activity that provides you the transport of these hydrogen atoms from one space to another. So all these little workers are hydrogen atoms, and they're going to use the electrons from NADH to move from that mitochondrial matrix into the intermembrane space. So this is where we have all of these protons being moved via this electron transport system. Now, what's going to happen is that you're going to build up all these protons in this intermembrane space over here, as more and more of them are moved via these electrons from NADH and FADH2. Now, these protons, once they build up, eventually they're going to return back to the mitochondrial matrix, the innermost part of the mitochondria. When they come back, it's like little workers, they go off to do their work, and when they come back, they earn money. So they earn money, and what's the money? Their reward is ATP. So when they come back, their final reward is in the form of this energy currency. And that's the whole purpose of this. We want to generate ATP, so the more protons you have moving in that space, the more that can come back and feed that ATP synthase that drives the formation of ATP. Alright, so there's a cellular respiration debate. Now, do I want to participate in this debate? Not really. So how much ATP is produced per glucose? And Martini and also they say 30 to 32 ATP per glucose. OpenStax and older biology and chemistry textbooks, they say 36 to 38. Now, where does this break down? There's a debate surrounding how much NADH and FADH2 is converted per NADH and FADH2 molecule, how much ATP you get from that. So do I really care about that? Not really. Again, and I want you to take this to other classes. This is only for this class. I'm not going to say like, put these like, okay, how much ATP do you generate per glucose molecule? The answers are 30, 32, 36, or 38. I won't do that to you for this class. If I do test you on it, it's going to be a ballpark thing, and the other answers are going to be obviously wrong. But does this really matter? Let's take a step back. So what I want you to know is like, okay, glycolysis, this produces 2 net ATP, because why? You put 2 in, but you got 4 out, so 4 minus 2 is 2. Citric acid cycle produces 2 ATP per glucose, and why is that? Well, again, you have pyruvate, so you have 2 pyruvate from 1 glucose molecule. Each pyruvate is going to generate 1 GTP molecule that's converted to ATP. So if you have 2 pyruvate, you're going to end up with 2 ATP. So that's why that's the net from citric acid cycle. So yeah, that's why I outlined here. Now what you're going to generate is the major product from the citric acid cycle. The citric acid cycle is important because it generates all that NADH and FADH2. Now with the electron transport chain, whether it's 30, 32, 36, or 38, it generates the vast majority of ATP directly. So it's going to take all the NADH and FADH2. So my analogy is like you're taking all this built up NADH and FADH2, now you're going to cash it out in the form of ATP. So this produces the majority of the ATP during cellular respiration, that big equation where you take glucose, oxygen, combine it to make carbon dioxide and water. So this is the big picture right here. Notice that the electron transport chain generates the vast majority of ATP per glucose molecule. So you have to go through all these steps to finally get the NADH and FADH2 that produces all this ATP from aerobic respiration. Alright, so this is what I want you to know from this, is that, and this is why we have this equation right here. So what we did is take the glucose, we did glycolysis, we broke it down into pyruvate, and from that pyruvate we lopped off the carbon dioxide, so that's one source of carbon dioxide. We made acetyl-CoA, we fed acetyl-CoA into the citric acid cycle. The citric acid cycle also generates carbon dioxide as well, and a tiny bit of ATP. But the major product of the citric acid cycle is NADH and FADH2. That NADH and FADH2 ends up in the electron transport system. That is going to use electrons to move protons between the different membranes of the mitochondria. And those protons are finally used to make ATP via ATP synthase. That's the overall, this is what this equation sums up right there. Okay, so this is what I want to talk, and this might be a review to those of you who already took AP Bio, but this, especially that citric acid cycle, it also comes into that acetyl-CoA is very important when we talk about how to metabolize and convert fatty acids and amino acids into energy. And we'll have to save that for Wednesday.